Thank you very much. It's very good to be here, and I hope that I can share with you what we've been doing in the research world with BDFA for the last few years. And what I really just want to start to do is to set this, I really feel that I have to set in context the disease that for the families affected that we represent. What's Batten disease? Well, Batten disease, the official name is neuronal ceroid lipofuscinosis, which is quite a mouthful, the MCLs. And there are several different genetic life-limiting neurodegenerative diseases. They were first identified by a scientist at the beginning of a clinician really, at the beginning of the century, and that's why it was Frederick Batten, and that's why it's called Batten disease. Now these are simple genetic diseases. That's probably the only, simple thing about them. And this is a schematic from, we produce lots of support leaflets as well. So 2 unaffected carriers, that could be any of us. You can be a carrier, which means you're unaffected because you have 2 copies of the gene, and therefore if you only have a fault in one of them, then you will be fine. You will not know you're a carrier, but sadly if 2 carriers are together, then there's a 1 in 4 chance with each pregnancy that a child can be affected. And there are different types of Batten disease, and this is one of the challenges that we face because there are actually 14 different types identified genetically at the moment, and I'm sure there will be more because there's a great deal of actually research going into identifying the genetic causes of Batten disease. And they're all called, the genes are actually referred as CLN, and there's a whole variety here. For instance, 1 and 2 mean that you're missing a very vital enzyme. Several are you're missing a protein which is in the membrane of a particular part in your cell. So these are devastating. The effects of this are really quite devastating. So, and also we have a lot of different, they have the same symptoms, the same outcome, which is children suffer from epilepsy, and sadly premature death is inevitable. But actually the functions of these are different. So when we're talking about, we were developing our research strategies, I'll come on to say, there were different questions that need to be answered and still need to be answered for each of these. So we have quite a challenge in how we represent this. So as I've just alluded to, the problem is all of these are located in the lysosome, which is an organelle within all cells. And what happens is your lysosomes are responsible for the breakdown really and recycling of waste, and when this fails you get a lot of build-up of material, and one key set of cells which really can't cope with this very well are nerve cells in our brain, neuronal damage. That's why it's termed a neurodegenerative disease. But I'd like you to sort of also bear in mind when we're talking about research is that every cell in the body is missing these proteins. So this is the challenge, and there are a lot of questions that still need to be answered in Batten disease research, and maybe in the organisations and the diseases that you represent. There's still a lot of fundamental questions to be answered. So the BDFA was formed in 1998 by a group of affected parents, and Pauline here, she has 2 children with one of the forms of MCL which occurs, the onset is slightly later. This is often referred to as juvenile MCL. Sadly, her daughter passed away. She still has a son, which is still part of our board. And Irina has a daughter affected by the same type, and Rachel here and Julie, both their children were affected by what's often called late infantile. Their children, the younger, the missing enzyme form of the disease. So they formed the Batten Disease Family Association, and where have we gone from there? Well, now we're an organisation. We have, this is our Chief Executive, Andrea West, Lucy does office management, it's myself. We have fundraisers, and we have a support officer. Barbara is retired, but she's our education advisor. And we're very, very dependent on our supporters for fundraising. All our funds, everything that we do comes from our fundraising base. We have a great a lot of volunteers. This is our latest social media volunteer. She's the one who was getting me to tweet, which for me has been quite a new experience. And here, researchers. We really do interact with researchers. This is the key. We do a great deal of support work. Harriet says our family support officer. The here and now for our family support is key. But research is the future. This is the hope. This is where we have to go, what we have to invest in. And this is the newest group that I've been visiting. They're actually from the Roslin Institute in Edinburgh. And if I say to you Dolly the sheep, then you'll remember what you imagine. They're working to look at large animal models of disease, and they are now very interested in Batten disease research. So as I said, we have our three aims. We want to support families and the professionals who work with them. You know, we believe that no family should go on this devastating journey alone. We raise awareness. This is the Spinnaker Tower near actually where I live. And we have a Batten Disease Awareness Day and we turned it orange. The key, what I really want to talk to you today and to share with you is our experiences of directly funding research. So the board set a research strategy in sort of 2007-8 because initially we only had the funding for support. That was the key thing, and in a way you have to support, you have to fund fundraising to start. And then as our charity grew, as our funding grew, then the board set a research strategy. And this is quite an overarching strategy, and I hope you understand that from the nature of the disease and what we're dealing with. But we wanted to, we have, you know, to support all our families. We fund research into all forms of the NCL, excellent in research and peer review, as you just heard, is very, very important and crucial to the work that we have funded. Partnerships are key. We wanted to be very proactive with our stakeholders, and that means the researchers that we knew were doing the work, other funding bodies such as, for instance, the larger bodies that you've already heard about, who also fund research. And we knew that we'd probably have to be innovative, because when you're dealing with a rare disease that's been brought out in the previous talk, sometimes you do have to say, well, actually things are just not moving fast enough. And we need to say that actually this is where, from our experience of our families, what they're telling us is critical and important to them, and from our expert advice, this is where we wish to go with our funding. And obviously there we have individual families who they get a diagnosis, for instance, in the rare forms of the NCL, some of them are really ultra rare, there may only be 2 or 3 affected children in the country. They've often independently raised funds and they said, well, we would like you to help us fund research. And that can be a challenge, and one of those I will come on to talk about later, because how much funding they raise can be very variable, in a year and what they want to do, and how best to spend what can often be smaller amounts of money. So within that strategy, and I'm sure you've all seen this similar before, we want to go from basic research to treatments. And this is really a summary that I put together actually for our annual general meeting as a visual picture for our families of what we're funding, what we've achieved. And really what I want to sort of talk about today are examples to show you the way we work, because I think that's more powerful. So I'm going to talk about this example here, which is part of collaborative funding and family funding, here to do with funding and drug repurposing and screening. This, which has been a very successful project looking at treating visual failure, because blindness is one of the first symptoms, and obviously when you lose your sight, that's a huge impact on quality of life. And our families early on said to us, this is a key thing that we want research into, because many of the children are actually quite well for quite a while if their seizures can be managed. And when you lose your sight, you also then lose the potential for attending in more mainstream education for longer, all these things. So this was a key priority. And we are now, there is now one treatment for one of the, which is a missing enzyme, so enzyme replacement therapy is relevant. And we do work with pharmaceutical companies, but it's more at the moment at the end stage on the support issues when there is actually things to go into a trial. So the first thing I would just like to talk about is one that's very, very important, and we're working with researchers, is on a patient registry. In a rare disease, especially when you're running a clinical trial, the control arm, if you like, is going to be against the natural history or against data that has been collected from patients over a number of years. And this really, in rare diseases, it needs to be a global initiative. And I would definitely say here, that this is where patient organisations can drive and promote researchers collaborating. Because, as I've told you, in some of the ultra-rare diseases, there may be only 2 or 3 cases per country. So we need significant amounts of data. And I would like to use this as one of our examples. It's not just a question of funding. And yes, you'll see from here, we have put in funding. This was initially funded by the European Union grant through the called DEMCHILD, which Angela Schultz applied for, and this was 3 years of funding to really start this. She had in Germany a very good registry, and she applied for funding to expand this so that data from other countries
could be added to her database. Now obviously part of the cost of that, as you know, there's ethical approvals, quite rightly, say in the UK, for data to be added to another database, and Ruth Williams is the clinician in the UK. So once this grant had finished, this has been set up, but then it's maintaining it, and we have partnered with the American Batten Disease Research and Support Association, and Noah's Hope and Hope for Bridget are two individual foundations also based in the US, and families with children affected by CLN2 and the BDFA. And so we have partnered to provide funding for actually a data analyzer, because there's a lot of data in there and you need to analyse this for it to be useful. But another way that we've really been partnering with Angela and with Ruth is actually, this is fine to set this up, but what we need is for patients to say they are willing, families willing, for their children's data to be included in the registry. And one way that we've worked with researchers here is that I always have this slide wherever I go, whenever I talk to families. We always say to families, this is your personal choice. We would just like to tell you how important we feel this is, and we want to support you to take part if that's what we wish to do. So this is an example of, it's not just about funding in rare diseases, sometimes it's about the actual support you can put in. And this is progressing well, and also it's the patient organisations who are moving forward and actually getting other countries, such as in the US, where they have a large amount of data, to meetings together to say, how can we make this a really global initiative? And so the other thing that we do, funding, which was what we did first of all when we had, say, grants of £5,000 to give, was we partnered with universities. I've used UCL here as an example because this is the first one that we did. We partnered for a studentship, and I don't know how much of you know, but essentially for a student you need a stipend, they need to be paid because they have to live and eat as well as do their work. But often universities or other funders will provide this, but they might not provide fees or travel or training, for instance, travel for them to attend meetings. And that's very key, I think, especially for young researchers to know and to interact with other researchers. Or often this thing, which is often just referred to as consumables, it really means the things that are used in the laboratory. And sometimes in rare diseases and in Batten diseases, a lot of the work does involve the maintenance of mouse colonies, and this is very, very expensive. And we were finding that often studentships were not progressing as well as they should. And this was a good way initially when we had small amounts of money to do this, and obviously all the peer review had already been done by the university or somebody else. Because for £5,000, and initially we didn't have the staff or the resources, running a full programme as the ones we've just heard about is not practical. And this is just a sort of summary of what we did, and because I do want to talk about the fact what worked, what didn't work, these have all worked very well. This one didn't mean that it was meriting less funding, but this was also competitive. And it was, the researchers had to submit what they wanted to do, why they needed this. And actually this product did not require as much funding as the others. And so we partnered with all the different partners here, and this is a German research Batten Foundation that funds, and I told you about the American one. And this here was actually the one where the project was very highly reviewed, had a very high score, but actually, and the researcher was the principal investigator was very honest. After the first year he said, this is really not yielding the results we hope, this is not going in the direction we want, it isn't something that is fair to the PhD student, she's likely not to get a PhD, not through any fault of her own, and that he said to the joint funders, in that case this was us and the NCL Stifter, we don't really recommend, we're not applying for further funding. So it doesn't, always work, but predominantly for us it was a very good method of funding and getting high quality research, and research which moved many, many important projects forward for a little administrative effort on our part. And it had other important benefits for us. For instance, we could start a programme of lab days where we said, well, we're funding your student, so we'd like to come and invite our families. They need to see firsthand what these students are doing. And we had some fantastic days. So this is the one at UCL, and here he is. Davide was one of the students, and the students, in fact, many of them still follow what we do. for them, they were slightly daunted, I think, about meeting families, but all of them said it was a positive experience, and they said it actually really brought home to me why I am doing what I am doing. And I would say that we always make sure that myself, I'm usually there, and we do have members of staff, because sometimes it can be daunting for students, and you do need to provide a level of protection, really, is the only way I can say it, because sometimes the family might say, you know, my child is dying, now how is this really helping? And that can be quite a hard question for a student to answer. And obviously their heads of labs are more used to that. So I would recommend that this is a great thing to do if you get to this stage, but I do think you need, again, to be respectful of that. But we've had some fantastic days, and also it was a benefit because we could ask other experts at each institute, come along, you have some expertise, we think you could be of value to the field. Please come and talk, or come and listen, come and see. And in fact, there were some very good collaborations that came out of some of these lab days. For, again, very little cost and effort. And more importantly to me, it allowed our families to see exactly, as we heard before, where our funding is going to. And we're still doing this. This is Batten. This is Freddie. I'd like to introduce you to Freddie and Louis. And they're Batten, and they're CLN5, which is twin boys with a very rare form of an NCL. And they raised a lot of money, and actually, but what has allowed their funding to do is that Cardiff University, again, this is all peer reviewed, and the project wasn't chosen because it was match funding, but actually this project scored very, very highly, and therefore the university are putting a great deal in, and the contribution there is significant. It means there's a 3-year project. And we still don't know what this protein does. So, uncovering fundamental differences is a key question. And that's Amir, and that's Katie, the student. Now, the other thing you can do is, I think there's representative from Sparks here, you can partner with a larger charity, because often larger charities will do the peer review process for you. And this is something we did because we wanted to do a 1-year fixed project. And so Sparks did the review, the call for us and the review process, and they scored the proposals. But it was the decision of the BDFA and the BDFA board that provided anything scored to be high enough fundable, then that decision would be for the board to make. And in fact, there were 4 projects which actually scored quite closely, 2 of which were considered to be outstanding. And the one that was chosen was this one, which is for a research assistant, and this is Anna. And this is in Professor Cooper's lab at the King's College. This is the Paediatric Storage Disorders lab. And and they do a lot of analysis on animal models, what is actually going wrong in the brain. And when you treat animals, what is being affected first? What are the key processes? Because sometimes we can make things better, but is that really going to affect the disease? And in New Zealand, they have actually a naturally occurring sheep model of CLN5 and CLN6. And so it's very, very important as well. We felt there was a missing point of getting some expertise, somebody trained who really could understand and do this work and this analysis. So I think this is a key project, and this will also maybe lead into, I told you just briefly at the beginning about a group that is interested in doing more large animal work. So we also fund projects where we see there's a clear need and as much collaboration across different projects. And so Anna's been in New Zealand actually learning about the techniques, and will finish at King's her project. And that, I think, is a very, very important project. And this is drug purposing in Batten Disease. This is fishing for a cure. And I'm not going to, this was a lightning talk that I did at another Find a Cure meeting. I'm not going to go through all of this again. But this is about drug repurposing. Now, we hear a lot about drug repurposing. We hear a lot about drug screens. I've sat through many meetings, especially perhaps with people from large pharmaceutical companies, and I don't wish to cause any offence here, who say, you've got to screen 50,000 drugs. If you don't screen that, you're not going to find things. But actually, I've heard other people in rare diseases say, actually, a high content screen, which means where you're screening fewer drugs, but you're looking for a very specific thing that is very relevant to your disease, is very important. Now, this is about, from this, and this is a lovely picture. This is Katie Freeman. Now, she passed away when she was 6 years old from the form of Batten disease where you're missing an enzyme. And her parents her parents here are Katie and Martin Freeman, and they, in her memory, they raised money for research. And for them, I think for families perhaps sadly whose child has passed away, for them it was to fund something which was a legacy. And they were actually quite keen to fund something perhaps that was like a piece of equipment. And actually at the same time, this is Dr. Claire Russell from the Royal Veterinary College, and she had developed a zebrafish model. Now, I don't know if you know why I use zebrafish. Some of you may have seen zebrafish, you can keep them. But the power of zebrafish is that you can produce a great deal of them. And they're very good in certain things. You can study very well in them. And I think, you can see here, this, I think we can just see with the naked eye. This is the wild type. This is one that's not affected. Here are ones that are affected. So you can see this, and you can study their movement. And also they mimic, if you like, seizures. So seizures are a key problem and a key indicator of disease. So, and she'd actually had several projects which had been peer-reviewed and successfully reviewed, but as it was explained, like the MRC, there's often only a certain number of projects that can be funded. They're all deemed fundable, they don't get funding. And Claire, I think, was struggling because one of the things she wanted to do was to use this very powerful model, and I think, again, you can really clearly see the difference. This is very early on, wild-type little baby zebrafish, they're just larvae really, they don't look like zebrafish. They don't move a great deal, but ones that are affected are already showing the sort of seizure-like activity. But she didn't have an automated way, the latest system for monitoring this, and I think this was hindering her applications. And we obviously had expert advice, we spoke to her, we spoke to the Freeman family, and they were very pleased to fund. This is a piece of equipment which will do this, which you can put them in 96 well plates, and you can do a huge lot of screening, and then you can screen different drugs, and you can look at this activity. And also with funding with families, we, if you like, hold, we don't necessarily close a review process, because it may be that we suddenly get funding, and we did, in memory of another child, anonymously, more funding. It was only for CLM to do research. So this fitted very well, because we were able to look at one of the projects that Claire already had submitted and say, actually, from that, we can use that and base a project on this, again, always with independent, impartial advice. I will really always stress that. Ginny worked for 9 months, and she really did an amazing thing in that short time and demonstrated that if you design a screen well, you can really get some very significant results. So this is just schematic, and it's really to show you that she's using the locomotion tracking thing, and she screened 556 compounds, and one has actually remained positive in that. And I don't really want to have too much science in the talk, but it's really just to say that it's having a positive effect on the key things that I told you about. And this is one reason I applied and was very pleased to present it at the Find a Cure talk. And that's another thing I want to bring in, because I knew that audience there would be people who would be very valuable contacts. And we have, Claire's actually made 6 or 7 very valuable contacts with industry, with other people who should be talking about taking this forward. Because further funding is going to be needed to see whether this compound is effective, say, in the mouse model. There is actually a dog model as well, but that would again be something probably to be funded by a company to see whether this comes forward. And also it may and should help us maybe find out about the mechanism of action as well. So now I'd like to go on to the final example to say is that Andrew Strauss, the former England cricket captain, is our patron, because he's a personal friend of one of our trustees whose daughter is affected by Batten disease. And in 2010 he did a very, very big fundraising effort at Lord's, and sufficient money was raised with that and another fundraising effort we had to fund a PhD studentship. And we had not expected to be able to do that so soon, and this was one of the aims that we wanted to fund a whole project. So this was obviously a peer review process that we did run ourselves with expert advice. We do have a scientific advisory board and panel. And the project that was chosen for the studentship was looking at gene therapy to the eye.
Gene therapy, as you know, if you have a genetic disease and you're missing a gene, gene therapy is the idea that you put a functioning copy, a copy that works, into cells, and therefore this will then produce the protein, the thing that you need, which is missing and not working in the disease. So this was a very important project. And also a little bit of history to this, in that another thing that we'd funded, which I think was starting out, is to say perhaps run an expert meeting. Because for a very small cost, similar as the workshops that we're at today, we ran an expert meeting where we invited people who were already in the field, and this is she's now become Professor Sarah Mole in the last week, Sarah Mole, and she invited Robin Alley, who's an expert on gene therapy, to the eye. So that's how he first became interested in Batten disease. It's something that was run by the BDFA, and therefore when we put out this call, he applied. And so this was chosen, and Sophia was the student, and just to show you that this is how they do it. And to cut a very, very long story short, she worked very, very hard. She had to characterise the system. This was done in a mouse model that had never been done before. The Institute of Ophthalmology, where Robin Alley has his lab and where he was based, they were very, very supportive, because she was the only person working on Batten disease. She worked very hard, and they'd had great success with other diseases. But essentially, there are certain types of cell within the eye that they treat, and usually that restores the vision. But as often happens, and we've discovered very much in the NCLs, it's never what you think. And although they could treat those cells, they could do exactly what they've done in other diseases, it wasn't having the effect that it should have. And at that point, this is something that was critical, because she had enough data for her PhD. It was at a very critical point. And this is where I would make a real case. I think continuity of funding is really important. Because if we had stopped funding this project at this point, then we wouldn't have got to the very exciting results. And that is something, again, it went back to a shorter review process. But the general feeling was that so much would be lost if this work stopped. The expertise of Sophia. And the Institute of Ophthalmology were very honest. They said, well, if we don't have a person doing this work, then all the other things that we can provide for you, which are many costs, which we will just provide, we're happy to do that, we can't provide. So I think that was very, very important. And sometimes, again, I think as an organisation, you do need perhaps to take a risk. And the risk was taken, and again, I've just put this up because I think these are really fantastic. And Robin was so impressed that he's now got pictures of these in his office, because he said this is one of the most remarkable sets of staining that he's seen. But fundamentally, it was you have to treat a different type of cell in the eye. And I just want to show you this, because I think again you can see this is untreated. This is very, very preliminary results. And I'd just like to make one quick point here. You do have to be respectful to researchers and be careful when you're talking about protecting the work. If it's unpublished data and they allow you to talk about it, then please be respectful of that, because also there could be intellectual property issues later on. You do not want to ever jeopardise something that might have to be patented, because a company, maybe if they want to take this therapy on, may need to look at that. That would be possible. Now this is got to be repeated, and this is very preliminary data, but we wouldn't have got there. If we'd have stopped the funding, we wouldn't have got there. Part of the criteria we also set is that at that time further funding had to be applied for, because this project we couldn't keep funding it, and also for success, plan for success, but then you're going to need further funding if you're going to expand this. And also, if it's successful, we want to know how are you going to expand this to the other types of Batten disease. They may be the same, they may be different. It may be different what you have to treat in the eye for the different types. And in between all of this, and this is probably quite topical for today, we were also invited to be part of, and this project went into this, a call by the European Union for rare disease funding. And I think on the strength of the relationships that we built with researchers, this required a patient organisation to be part of this here, or down here, because the key to this is a patient organisation informing the process. What is important to families? What do they want?
is research translatable. This is heading for therapies. Everything you do in this has to head towards a preclinical to a therapy. And the eye therapy work, because we funded it for that extra year, was in a very strong position to make it be a very big part of this application. And actually the total amount given, this is now 13 organisations, 8 different countries, including the BDFA. I'm actually the work package lead for that. That was a very steep learning curve, is a steep learning curve for me to be part of the grant. This is €6 million. This is huge for Batten disease research. And in fact, when I was talking to the person who did it, I said, I don't, we can't take the credit. And he actually said, as an organisation, we should take some of the credit. And all of you, whatever you do in research, you should take the credit, because it's often being brave and saying, this is what we want to do that makes the difference. And it's also allowed us to employ Laura to do our part of it. And so really just to kind of finish, it means that from an initial outlay for us, a huge amount of money has now gone into research on that project and into Bacchio. So really, as we said, excellence in science, partnerships, we are proactive, we're innovative, we use a range of different resources, and we do provide a way for families to fund research. And these again are all the people who have contributed greatly and have put in far more than our funding has really paid for. And finally, I do have to finish, because these are the people that, this is why we do what we do. And this is some lovely photos from our family conference, and our families do amazing things. Thank you very much.